So we have nicotinamide adenine dinucleotide, or NAD, NAD+, okay? Here's its oxidized form, here's its reduced form, okay? It's a very important molecule, like I said, for cellular energy transduction when it gets reduced to NADH, okay? And that's what this is what that basic reaction would look like. Going from the reduced form, we're adding hydrogen, we're adding electrons. So kind of note, the proton and the electrons are not actually existing as two separate species, but rather as a hydride ion, so sort of a hydrogen with an extra electron on it. It's just drawn like this in the book, sort of for clarity, so you can really see the electrons, since we've been talking about electron transfer. Here's the structure of NAD+. Again, I have no interest in you memorizing this structure. I don't even have interest in you memorizing these names, right? So there's kind of two sides to the structure. There's an AMP side here, which doesn't actually participate in the redox at all. And then there's the side that actually this nicotinamide mononucleotide, or NMN, which is the portion of the molecule that's actually undergoing the redox, okay? So here we have our oxidized form, and then when we put a hydrogen here, we have now a reduced form. And like I said, there are a lot of really nice YouTube videos. If you search like NAD+, or NAD, NADH in YouTube, there's a lot of good example videos. Many of them emphasize the structure because they're meant for biochemistry, where you'll probably deal with it in more detail. But since we're just trying to get ourselves to the free energy piece, you know, I just would say, like, watch with that in mind. Don't hyper-focus on it, because I'm not going to ask you to memorize this. What I will say would be important is that you saw a picture of a molecule, and one was clearly oxidized and one was clearly reduced. And it was a molecule you hadn't seen before. You could tell the difference between an oxidized and reduced form simply because one's going to have hydrogen on it, or one isn't, right? Like, you could look at a brand new molecule and know the definition of these things. Right? That's more important to me. And then down here, another way you can search for it, and that you'll see it a lot, is this group, the AMP group, could be phosphorylated. So you might see it as NADP and NADPH. It can be in a phosphorylated or unphosphorylated form, okay, the NAD. Okay. So this is a reduction half reaction, okay? So the NAD is getting reduced, but it's going to be paired to the oxidation of something else, right? Some other molecule, some other substrate, okay? Mediated through an enzyme. There'll be some enzyme that'll bind both the substrate that we want to oxidize and the NAD. It'll do the redox, and then they'll unbind from the enzyme, okay? The enzyme is a cofactor facilitating that transfer. And this is a really short, like 3-minute video that just shows that. It's nice, it has, just has some audio over the top, and it shows the enzyme comes in, the NAD binds, the substrate binds, it pops over the hydrogen, and then they both dissociate, okay? So I recommend that one, it's just really straightforward. Any questions on this? So one example of an enzyme that binds NAD for the purpose of oxidizing some other substrate is alcohol dehydrogenase. Okay? So it's an enzyme that will bind NAD in order to catalyze the oxidation of alcohols to aldehydes. So this is the process in your body when you consume ethanol. Not that any of you do, but if you did, this is what would happen. So here, the alcohol dehydrogenase is this 2-subunit green and purple molecule, okay? It has 2 identical subunits, both of which can bind an NAD. And this is just a close-up of that, okay? So it has a binding site for NAD, and then also in that same binding site in close proximity, there is the group that is to be oxidized, okay? So in this case, it's a benzyl alcohol. And so then this is sort of showing you what's happening in that reactive site. We have our oxidized form of NAD becoming reduced. We have a reduced form of benzyl alcohol, and it's becoming, when it gets oxidized, it's now benzyl aldehyde. Okay, again, I don't want you to memorize these structures. I do want you to recognize, if you saw this situation going without these words on. You could label them because you could see whether the proton is coming on or off and what's going on, right? So this is another video that talks about ethanol metabolism in more detail. This is just a screenshot that I pulled from it. The relevant part for you is 150 to 5 minutes, minute 50 to 5. Again, he does a good job of talking about this. I will just say, like, the bit before and the bit after is just more sophisticated than what we're going to talk about in this class, so there's just no need. But essentially, you consume some ethanol. Here's our alcohol dehydrogenase molecule, okay? It's a reversible situation, okay? So this is a reversible reaction where we can, what am I saying? Oxidize to our aldehyde. And again, it requires NAD to facilitate this. Now, once it becomes an acetaldehyde, there is now acetaldehyde dehydrogenase. Okay? Instead of alcohol dehydrogenase, now we have the aldehyde dehydrogenase, and it's going to function, it's in the mitochondria, so that's what that is showing. It also functions by binding to NAD to facilitate this reaction. Okay? And then you'll have acetate. Now, if you have a buildup of acetaldehyde, if this part here isn't functioning, that's what basically leads to hangovers, and is also a deficiency in ALDH2, leads to sort of a flushing response. So again, he does a nice job explaining this in the video. I guess I'm assuming the voice is male. Maybe it's not. Anyways, I recommend watching it. I thought it was very interesting. Okay. Any questions? Okay. I had not assigned these sections 4 to 7 as reading. Okay. It's just more redox examples. Okay. So in section 0.4, it talks about oxidized flavin adenine dinucleotide, or FAD, which is derived from riboflavin. It's basically the same situation as NAD getting reduced, except for that it happens in 2 single electron reduction steps via an intermediate. Okay, so here we have our oxidized form of flavin, and then it's going to our reduced form, FADH2. All right. And then sections 5 to 7 talk about the role of both NAD and FAD in a couple different things: glucose oxidation, ATP production in the mitochondria, and in chloroplasts. I don't want you to memorize any of those. Biochemist, the glucose, like that whole citric acid cycle and all of that jazz, that's what biochem is for. That's why you get to take it. Yay. I don't want to do it here. But like I said, make sure that you would recognize a redox reaction if you saw it. Okay? You would recognize an electron transfer.